Good morning or afternoon, wherever you are, everybody. My name is Jack Dekkers, and I'm a faculty member in animal science at Iowa State University, and I'm one of the co-PIs of the Agriculture Genomes to Phenomes initiative funded by USDA NIFA. We're very appreciative of their funding of this effort, really a community building effort. And as part of that, these field days, this is already our 13th field day. We will talk about institutional collaborations or industry collaborations, but also collaborations between different disciplines. In particular, in this case, we'll talk about opportunities for collaboration with institutions and groups that come from more from the engineering side, that are instrumental for us to be able to do a lot of the work that we need to do to advance the AG2PI initiative. So we'll have two examples presenting today. One is an industry partner who is working to develop software for biologists, and one from academia, from engineering. And as just as a disclaimer, the opportunity for these two groups to present in this venue is in no means an endorsement of these groups by AG2PI or by NIFA, but they're given the opportunity just to give examples of the types of collaborations and the challenges behind those, and how they could be set up in other situations. So the first presentation will be by a company called Progeny Drone, and they'll talk about phenotyping in small plot agronomic field trials using software that they've developed to collect data to analyze data that has been collected by drones. They are a startup out of Purdue University several years ago, and they're, among others, they're collaborating with the Maize Genomes to Fields initiative, which is a public-private partnership that builds on publicly funded corn genome sequencing projects to develop approaches to understand the function of corn genes across different environments. And that project has been funded by the Iowa Corn Promotion Board, as well as USDA NIFA, NSF, and others. So they are an industry partner in this project, but I'll also talk about some other initiatives that they're involved with. As presenters from Progeny Drone, we have Anthony Hearst. He's the CEO and holds a PhD in agriculture and biological engineering from Purdue. And first, I'm going to turn it over to Edwin Riedel, who has a PhD from Cornell University, and he leads the business development efforts of Progeny Drone. So as usual, if you have questions, please put them in the chat. And if they are pertinent to what is being talked about at that time, I'll interrupt the speaker and ask it at that time. But otherwise, we will ask those questions at the end. So Edwin, the floor is yours.
Okay, thank you, Jack, for this opportunity. And thank you, Nicole and AG2PI, for the opportunity to speak to the audience, and also to everyone who's here from the US and around the world, and those who will be watching the webinar later in the recording because it's the middle of the night for them. Thank you to you as well in the future. So Anthony has the slide started, so that's good. Yes, we are Progeny Drone. That's the name of the company. And our software product is called Plot Phenix. I know there's a little confusion about that because we used to call this software Phoenix, our Progeny as well, but we had to change that. So it's Plot Phenix, and that's short for Plot Phenomics. Can I have the next slide, Anthony? So the original programming, which eventually became Plot Phenix, was written by Anthony while he was a PhD student at Purdue. He was flying fixed-wing UAVs to map field trials in Professor Katie Rainey's soybean breeding program. And I think that's still Anthony's LinkedIn picture launching one of those fixed-wing UAVs. And he realized that the existing tools for stitching orthomosaics were quite inadequate, and that he had a better idea about how to do it. And the resulting code that Anthony wrote was the core of an application submitted by Anthony and Katie to Purdue Foundry's Accelerator Startup Program. And they won that competition, and thus Progeny Drone, the company, was launched, selling software in 2019 under license from the Purdue Office of Technology Commercialization. I came on board in early 2020 after running into Katie at the NAPPN meeting in Tucson. I was introduced to Anthony and asked if I could help them out with the development of the market. So Anthony and I have been working almost 2 years together. We've only met that one time, and we work pretty closely over Zoom. Anthony, can I have the next slide? So there's already a brief introduction to what Genomes to Field does. They were one of our earliest adopters, and we interact closely with the group, with David Ertl of the Iowa Corn Promotion Board and the researchers who are in multiple states doing phenotyping of corn and analyzing the images with the software. So we work closely to understand and respond to the challenges faced by the users. We meet with them for troubleshooting sessions as needed, and we've also solicited formal feedback via David Ertl to understand the challenges and incorporate those suggestions as we update the software. And I believe several members of the group that are using Phenix with Genomes to Fields are with us today. And with that, I will turn it over to Anthony.
Thank you, Edwin. So the Genomes to Fields initiative is really a perfect example of high throughput phenotyping. They have replicated small plot field trials across the US, and they have thousands of plots per trial. They're collecting frequent RGB and multi-spectral imagery ranging from 0.5 centimeter to 2 centimeters per pixel with 70 to 80% overlap. And this results in thousands of images per flight date. And so to do this type of phenotyping, you really need bookkeeping and automated batch processing. Also, it's important to think about where that processing is done. For example, you could do centralized processing where you process all the data at one central location. And there's advantages to this. For example, you would theoretically only need one person with high performance computing and programming skills. And hopefully the outputs would all be consistently processed. But there are also disadvantages. For example, getting the necessary data and metadata to that location can be slow and error prone, especially if you're relying on network connections. And you just have a lot of image files to transfer. You have to explain all the flights and plot layouts and planting errors. And also you'll have less consistency and opportunities for quality control of that raw data. There's also the option of decentralized processing, where you process the data on site, ideally right after you finish the flight with your drone. And advantages to this are it would require less coordination among the different field sites. And ideally, there would be more opportunities for quality control. There are also disadvantages, like you might need multiple people with high performance computing and programming skills at each of those sites. And it's possible you could get less consistent processing from those sites overall. But what if you didn't need high performance computing or programming skills to quickly, automatically, and consistently batch process UAV imagery in a decentralized framework? So there's ways you could approach that. One of the simplest ways is if you organize your images into folders for each location that you flew. You would want to have something like one location and date and camera per folder. And you would want to be careful to avoid overwriting or corrupting any of your raw image files when you're organizing your images. And ideally, you could just use like a point and click software tool to do the batch processing of these folders. And it's possible to accurately stitch approximately 1 image per second on an off-the-shelf laptop. Maybe it would cost, around $1,000, something like a gaming laptop, using good flight plans and appropriate algorithms. And this can enable you to stitch your imagery right after the drone lands at the field's edge for quality control. Some important factors to make stitching easy and fast is doing your flight at constant altitude with a single grid flight plan, and removing images from takeoff and landing before you stitch. And also avoid flying too low. You want to try to stay well above 10 meters or about 40 feet whenever possible. Also, flying with the camera at nadir perspective makes stitching easier and faster. A gimbal is recommended, and capturing high overlap is extremely important, especially for agricultural terrain. So we recommend around 70%. Also, good camera settings go a very long way. For example, you would want a fast shutter speed to prevent motion blur in your imagery. And also, it really helps if your terrain is at least approximately flat. And fortunately, that's generally true for small plot field trials. Of course, there are exceptions. But then what about locations that contain multiple experiments? How do you batch process that? So the way you can set this up is by only stitching imagery in each folder once. So we think of stitching as a location-specific operation. And then multiple experiments in a single location can be designated by plot layout files inside of each folder. And then you just have your software run the remaining process steps to get to the plot level once per plot layout file in each folder. So that includes things like gridding plots, extracting them, and analyzing them. These are experiment-specific operations. And then your software can just save experiment outputs in separate folders inside each of your input folders to prevent any overwriting or mixing up data. So I'm going to show you a few examples today. This is just a quick view of the specifications on my laptop that I'm using. It has 16 gigabytes of RAM or memory, and it's running Windows 10. I have 6 cores on my laptop, which is pretty good. So yeah, basically this is a gaming laptop, but nothing more. So first, as an example of how this kind of batch processing framework can be quite powerful, here I show us stitching 21 flight dates using the software we provide called Phenix. You can just simply select all of those folders by clicking and dragging, and then you can input a name for the location and the experiment in those folders, and you just check the box for the stitch step and you hit start. And in this example, we're able to stitch all 21 flight dates, which is about 4,300 images in about 98 minutes. So that gets to around 1.3 seconds per image. It's going a little slower because I'm doing screen capture at the same time. But then you can look at the orthomosaics from each flight date and see that they are stitched well. Now, there are some flight dates here where it appears they had some missing imagery, or maybe the altitude wasn't consistent. And that's a reason why having really fast stitching at the field's edge is really important for quality control. If they had access to this tool, they could have done the stitching right after the flight and immediately recognized that they missed, half the field on one of these dates. There are some weaknesses to this framework though. So first off, each camera only has one SD card. And so very quickly, your imagery can get scattered across multiple folders and the images will have duplicate file names. And if you want to do this kind of batch processing, you need to manually rename those images uniquely sometimes and organize them into separate folders. So that can be slow, tedious, and error prone. And if you make mistakes, it can lead to overwritten or corrupt image files, which can cause processing bottlenecks. There are additional challenges when you're dealing with really large experiments, like in the Genomes to Fields initiative. So basically, gridding orthomosaics into plots can be laborious. You have planter drift from row to row and planting errors, Also, stitching of agricultural terrain is basically never perfect because it's so homogeneous. There's usually going to be small mistakes here and there. And then also these small planting and stitching errors add up over large distances. So it makes it really hard to fit just a simple uniform grid over your plots. You need something more flexible than that. On top of that, because the imagery is so big, it's difficult to fit it on the screen to begin with, and that can make it hard to find each experiment in your field and keep track of plot labeling. And it also is difficult to precisely align all of those thousands of plots on every flight date. And so kind of the most common approach today for handling this is relying on really precise GPS coordinates, usually RTK GPS, as well as ground control points or GCPs, and perfect stitching and plot shape files. But there are problems with this approach. For example, getting RTK GPS on a UAV is very expensive, and also setting up ground control points, marking them in the images, and creating shape files can also be expensive, complicated, and time-consuming. And on top of that, even after you do all of this, the stitching will still probably not come out perfect just because it's homogeneous agricultural terrain. But what if you could reduce these large experiments into more manageable pieces? So you could grid them a section at a time in a manner that makes it easy to fit them on your screen and overlay your grids, keep track of labeling, and accurately align all of your plots. So this is a challenge we've been struggling to make easier over time. So here's a quick example of some of these aspects of the workflow and how we've dealt with these challenges so far. So I've got actually one flight date from a camera. It's a large field. And you can see in each of these folders, there's actually 999 images, and they have duplicate file names, so it'd be easy to accidentally overwrite them. So what we just added in the latest version of Phoenix is this tool that lets you select your folders with your imagery and just press a button that says merge folders. What that will do, it automatically uniquely rename all the images in those folders and put them together into one folder for that stitchable location. And in this case, they just happen to have a couple of extra images in there from a completely different location. And we might not have caught that unless we had a convenient tool like this, which just immediately said, oh, there's actually two folders for this data, not one. So it also will weed out corrupt images, so you don't have to worry about those blocking your workflow. After you've done that merge folder step, it's good practice to remove the images from takeoff and landing before you start stitching, just because they can mess with the accuracy. After you've done that, you can just click Add Experiment, put in your name for your location and experiment, check the box for the stitch step, and hit Start Processing. And again, this is an example of a pretty large data set, 6,540 images. But with the tools we've developed, it's possible to stitch this in just under 3 hours. And that amounts to around 1.5 seconds per image. Generally, we get closer to 1 second per image if your data set's like around 1,000 images. There's just some extra overhead with really large fields. Anyway, after you've stitched, then you get to your experiment specific steps like gridding. And in this case, we have a really large field, so it would be difficult to just try to fit one grid over the whole field because you've got those small stitching errors and planter drift adding up as you go across the mosaic. But with this workflow, you can actually break down that really large field into smaller, very manageable chunks. So for example, here, I'm just gridding the first 10 rows, and these are 4-row plots. We're also displaying the mosaic with like an enhanced color scheme so you can more easily see where the rows are. And then you just start by pinning down the corners of your section of the experiment, and you can adjust your units and your measurement zone. And we also have some feature called auto centering, which helps to get your plots aligned with the rows. But before you do that, you should first just quickly try to align your plots manually, just to give auto centering the best possible starting point and a good chance at succeeding. It's also very important that you have the correct sub-row spacing displayed in your plot layout panel, so the auto centering knows the spacing between the rows and can center properly. So once you've done that, you can get a quick preview of what your output plot images are going to look like after auto centering has been done. You can make adjustments there if needed, but usually you don't have to make a lot of adjustments. It depends on the field site. So then the next step is to go on to the next block of plots. You just change the name of the experiment in your plot layout file in that folder. And that tells Phenix, oh, now we're starting a new experiment. And then what you can do, this new feature we added is the ability to overlay a shapefile on the map. And Phenix actually creates a shapefile for every experiment that you grid. So you can just grid a block of plots. Then when you're ready to move on to the next block, you can overlay the shapefile from the previous block. And that just serves as a very nice placeholder kind of for where, so you can easily pick up where you left off when you move on to the next block of plots here. So you can see this field actually has 50 ranges going from bottom to top. And I'm ignoring the border ranges at the bottom and top end of the field. And I just split it into, I'll do the first 24 ranges going down the field. And then now I'm doing the second 24 ranges going down the field. Again, you just make manual adjustments where you can, and then take advantage of auto centering where it works. Generally, it works well when you have straight and narrow rows, but it doesn't always work. Like, for example, late season when you have high vegetation cover, it's more difficult. I'll talk more about that later. But yeah, essentially, by breaking down this large field into manageable chunks, you can work your way across the field without making any mistakes. And in this case, I was able to grid 3,000 plots in this field really accurately and reliably at about 1 plot per second. And so this is maybe the type of task that you could have an intern do for you. Doesn't have to require like a lot of technical expertise. So once you're finished gridding all of your sections or experiments, then you just copy paste your plot layout files, all of them, into your input folder before you run the remaining process steps. And that signals to Phenix, okay, it's going to process every plot layout file in this folder in batch mode. You can also, with the new version of Phenix, choose to generate just one image for every plot. You actually get multiple images of each plot in your flight, but we just pick the kind of the best image for each plot. And that can dramatically reduce the time it takes to generate your outputs and reduce the amount of storage space you need. So in this case, it took about 5 hours from start to finish to count 183,000 plants. So that's about 10 plants per second, which is a lot faster than most humans can count. Of course, that's not taking into account the time to do the flights, but it's at least as fast overall. And we're improving it from year to year. So once you've got your outputs for each experiment in a separate folder, you can then look at various diagrams just to double check that you have consistent labeling going across the field. You also have you have your plot images, which are extracted from your original raw photos. So these are going to be free of any stitching errors and lens distortion, things like that. You also can have things like measurement diagrams, so you can visualize the metrics, and it helps you to evaluate their quality, their accuracy. For example, here we show stand counts, where it will mark each plant with an individual plus sign that's inside your measurement zone. So you can look at those things at the plot level or for the whole experiment or block of plots, if you want to flip through it that way. And again, this is all just to help you verify that things were done the way you wanted. There's also color maps to help you visualize the variation in your measurements across your plots. So you can very quickly see if the ranges of the values make sense. And of course, you can get spreadsheets that have your metrics with the corresponding plot labels. And those spreadsheets can be exported into, other database software like ARM or things like that. So there are additional challenges when you're working with multispectral imagery. First off, we get images from different wavelengths from different cameras. It's not all through the same lens. And they also will have inconsistent sensor settings, lens warping, and fields of view. And the different camera makes and models may require different calibration procedures like MicaSense, DJI, Sequoia, Mapper, for example. And so we had to think, well, should we keep the images from the same camera rig in the same folder? How do we approach that? And when and how should we align the images from the different wavelengths? Because you have to do it at some point. And the traditional approach here is to align your orthomosaics into index maps. But there are some problems with the approach. Again, stitching agricultural terrain almost never comes out perfect. And that means your index maps will contain geometric and radiometric distortion, and that can lead to inaccurate index calculation. So you can sort of see in the image on the bottom left, it's that typical kind of blurry orthomosaic look. But what if you could instead merge your multispectral images, the original images from the flight, from each capture point into precisely aligned and spectrally calibrated multi-page TIFF files before you do stitching? So this is actually the approach we've implemented in the latest version of Phoenix. So here's another example data set provided by one of our collaborators at Genomes to Fields. And in this case, this is a typical output from a survey of a large field. You will get your imagery saved in multiple folders. It may have duplicate file names. There'll typically be thousands of them. In this case, I think there's almost 7,000 images across these folders. And again, with the new version of Phoenix, you can just select those folders and then press the Merge Folders button. And Phoenix will recognize, okay, this is a multispectral camera rig, so it should put all of these images in the same folder because ultimately we're going to merge them together. And it also uniquely renames them for you, so you don't have to worry about overwriting images or corrupted image files, things like that. So now that we've got them merged, the folders are merged, the next step is to merge the images from each capture point. So at that point, you just press the Merge Images button, and then Phoenix will create another output folder that has your merged images. So in this example, again, this is with a MicaSense Red Edge. Here's an example of a merged image of the calibration target. And again, in this case, what we're doing is we're accounting for the different lens distortion from each camera. We're doing spectral calibration on the images to remove the effects of different sensor settings. And you could see in that image from the flight that we get really accurate alignment across the wavelengths. It does take a little time. That merge images operation actually took about 45 minutes for this data set. So we're working on speeding that up, but it can be done in batch mode, and it's still overall makes the workflow much faster and simpler than it was in the past. So you can see here now what I'm doing is I'm putting the images of the calibration target in a separate folder, and then I'm also removing the images from takeoff and landing, anything that's not at like the main altitude of the flight. And that's just good practice. It's going to result in better, faster stitching. But again, what we're doing here is we're actually stitching the merged images. So now we've actually gone from, 7,000, almost 7,000 images to about 1,200. So you can see down there. And from this point on, the workflow is basically the same as working with RGB imagery. You typically need to set your image stitching scale to a finer scale because the imagery is lower resolution. You want to try to find a good balance there to get fast and accurate stitching. So in this case, it ended up taking about, 11 minutes to stitch this, 1,270 photos. So that was about half a second per image. So basically the processing time depends on the size of the imagery, the resolution. And there you can see we can generate a color composite orthomosaic from the merged stitched images, along with that kind of enhanced black and white mosaic to make gridding a little easier. And so now I'll show you what you can do. If you want to do the gridding step in batch mode, then you can first create your plot layout files for the different sections of your field, and then run the grid step, instead of just creating one and then changing its name. So basically, I start by gridding kind of the lower third of the field. You can notice that in this mosaic, there may be a little bit of warping. Like the rows in this field are probably pretty straight, but the overall mosaic has like a little bit of curvature. So if you tried to map one grid over the whole thing, it wouldn't line up very well. And so this is another situation where breaking it down into like 3 sections can make it a lot easier. Again, I'm gonna try to use auto centering here, but you can see that actually the auto centering did not work very well in this case. And that's mostly because it's much later season corn and there's some planting irregularities. But you can still manually adjust the plot images so they line up properly. That does take more time, but if you break up your large fields into manageable sections, you know, it all fits on the screen nicely and you can get it done. It definitely takes time though. And so we are working on ways to try to further automate this plot gridding process. Right now, we have this tool which, you know, lets you get the job done in a reasonable amount of time and works very reliably. But we're hoping, you know, over the next few years to add more features to make this even faster and smarter and less time consuming. But it's definitely still workable, practical, as long as you're careful about how you approach it. So yeah, here's just another example where I can manually click on these plot images and get them centered the way I want. And what that does is basically that tells Phenix exactly how you want your final plot images to look. And so it'll generate your final plot images at maximum resolution. So right here, we're just working with low resolution previews. You know, after you're done gridding, if you want to double check that your spacing makes sense, you can overlay all the shape files, just look over the whole field and make sure it looks realistic and accurate. And then you also, with multi-spectral imagery, need to do the step where you measure your calibration target. It's a little easier since we've merged the images of the target, but you also need to remember entering in the correct reflectance values for your particular calibration target. You can see I can read them off from the photo here. And that way your calibration will be as accurate as possible. So we have a little tool that helps you do that. You got to be careful with MicaSense panels. They don't list the values in order of increasing wavelength. So just watch out for that. You can also enter the serial number target for your target here, just for documentation purposes. And then once you do that little measurement, you just hit save and close. And then that basically signals to your software, oh, okay, I've got to generate reflectance imagery for the outputs. So then again, you can choose the option where you just create one image of each plot and you just you can check boxes for your measurements. You can choose from a range of options for indices. In this case, I'm doing NDVI. And then again, this last step is automated batch processing. So it knows there's multiple plot layout files in this folder, so it automatically processes each of them one at a time. And the last few steps are automated, so it just takes, I think, another 30 minutes or something like that here to get those metrics at the plot level. And then you get, I'm getting ahead of myself a little bit here. It's just finished the first section and now it's doing the second two. It's also generating some color maps to help you visualize the data. So I think it'll be finished with the last section in just a moment, and then we'll take a quick look at what outputs look like from multi-spectral imagery. Okay, so yeah, we got that all done in a little under 2 hours. And again, that's going from raw imagery to finished plot level images and metrics. So here's an example of what your plot images will look like with this workflow. You get multi-page TIFF plot images, and you can evaluate that. You can see that they are very accurately aligned. And that's because we did the alignment on the original images, and we're extracting these plot images from your original images. So we can maintain very accurate alignment and spectral calibration. And then you can visualize the metrics, for plots or for the whole block of plots. And you can look at color maps, make sure your metrics are looking reasonable. And then, of course, you get your spreadsheets as well, which can be exported to other database software or shared with the rest of your team, like if you're working in genomes to fields, for example. So, this workflow can help to make large data sets like this more manageable. It's certainly not perfect yet, but we are improving it from year to year, thanks to support that we get from people like Genomes to Fields. And here are some remaining challenges and areas of future work I'd like to address, hopefully this year. I think it's possible to do the image merging faster. To me, it seems like merging images should be faster than stitching them, but there's some details in the workflow that make it a little slower than you would think. And it needs to be perfect, otherwise it's worthless. Also, we need to exploit this image merging better, like offer custom indices, classification tools, machine learning, better visualization. So those are other things I hope to work on. We also really could use more robust auto centering so that people don't have to manually center all their plot boxes. It's not as easy as it might sound. Ideally, you could find an algorithm that works for all kinds of imagery, crop types, plot layouts, and growth stages. I have hope that that's possible, but it's a work in progress. And I hope this year I can do more for that. Also, we'd like to further simplify dealing with large or multiple experiments, give you like a little better control over which experiments you process and when, but without breaking this batch processing capability. So that's it for my presentation. Thank you. And we'll wrap up.
Okay, thank you very much, Anthony and Edwin. And thanks for leaving some time for questions. So put them in the chat if you can, or you can just speak up. But yeah, appreciate the presentation. Looks like very powerful software, very useful software. And as we're waiting for questions, maybe I'll ask one here. So what do you see as the major challenges in working with the researchers? Of course, I'd imagine they have lots of requests. So what are the challenges in terms of determining what they need and how you can accommodate that?
So it's, every project and every researcher has different needs. I mean, there's some commonalities, but we have to try our best to provide tools that kind of work in a lot of scenarios. And we also, we struggle still to try to reach out to our partners and check on how things are going for them. I wish like I could do a better job at encouraging people to sort of let us know when they run into challenges. So there's been like a few cases, not with Genomes to Fields, of course, but where we didn't hear from one of our users for a while, and we thought they were happy, but then when it got time to winter time, and they at that point started analyzing their data and noticed a problem, they were very unhappy. So and it's also a little tricky, especially if you're kind of doing the development of the software. You have to not take critical feedback personally, and that can be hard sometimes, but it's absolutely necessary if you want to survive in industry. So I guess I wish we had a better way to get more feedback from our users.
That's something we're definitely working on. It's like small company, shoestring startup kind of issues. We don't have a big staff yet, so we gotta grow carefully and try to cover all of our bases meanwhile.
Mm-hmm. Yeah, but I think that's a good message that for teams like this and for collaborations like this, two-way communication is key, right?
For sure.
Question in the chat from Chris Hansen. Are there any plans to support point cloud data, for example, LiDAR?
Thank you, Chris. So right now, we do actually have the capability to generate point clouds from the RGB imagery for individual plots. It is computationally intensive and a bit slow compared to some of the other metrics, but we do have that option. So we can generate point clouds. They're in PLY format, but we currently don't have the capability to import point clouds from other sensors or software such as LiDAR. Ultimately, I have hope that you can do enough with point clouds from RGB imagery alone so that you might not need LiDAR for most applications, especially because LiDAR can be quite expensive and has its own processing challenges. But there may be some applications where LiDAR is probably the only way to do it. To be honest, I don't know if we don't have a specific plan to incorporate it right now. But if there's an application where it's really, it makes sense. And especially if we have users who want to try things like that, we do our best to make this tool work for you.
Hey, there's a question from David Erdl. Says, if we use this software, do we have to realign the plot orthomosaics for every flight? Or can we do it once for the first flight and then it's set for the future?
That's a great question. So with this workflow, so if you don't have RTK GPS on your drone, then chances are that you will have to realign your grid on every flight date. So the way we handle that situation is on the first time you fly a field, you kind of have to draw the grid from scratch, like I showed in this presentation. But after that, the grid is saved in a plot layout file, among other formats like shape files. And then on future flights over that same location, what you can do is take that original plot layout file from your first flight date and you just copy paste it into your new folders of images of that location. And then Phenix will see that plot layout file. It'll recognize, okay, I should just reuse this old grid. So when you run the grid step, it takes that old grid and overlays it on the new orthomosaic. But again, the problem is if you don't have RTK GPS on the drone, then that grid from the previous orthomosaic will not line up perfectly with the new orthomosaic because they have slightly inconsistent GPS coordinates. But with the tool we provide for gridding, it's flexible enough so that you can usually just refix your corners just by dragging them over by a few meters. Occasionally, you'll have to make a few other adjustments to the plots in the grid to get them to line up with that particular orthomosaic. And that can certainly be tedious. That's why I was emphasizing the need for more robust auto centering of your plots. Ideally, maybe you could just fix those corner plots and then Phenix could just figure out exactly where your plots are between those corners. That's kind of somewhere I would like to get as soon as possible. But it's a work in progress.
Okay, thank you. And there are a couple of more questions in the chat, but we unfortunately have to move on. Maybe we can answer some of those at the end if there's time, or otherwise they can be answered online later on. So thanks, Anthony and Edwin, and you'll stay online, and so there may be some follow-up questions. So our next presenter is Dugan. He is an associate professor of mechanical engineering and geospatial computing science at Texas A&M University in Corpus Christi. And he's going to talk to us about AI-based AI-based plant phenotyping and harvesting for sustainable agriculture. And it sort of builds on the previous talk in that he's using the image analyses, but then also integrating that with robotics on the field and of course using artificial intelligence. So Dugan, floor is yours.
Great, thanks, Jack. It's my great pleasure to be here for everybody. So this is a kind of a collaborative research project we are doing with the Texas A&M Agri-Life Center. And then I'd like to share some of the recent findings and our research directions for the future. So if you have any questions, let us know, and then I can try to answer, okay? So the topic is AI-based plant phenotyping and harvesting for sustainable agriculture. So again, we have seen a lot of climate change, and also like COVID situation, we're losing a lot of laborers. So how do we deal with all this situation, mass situation? And we try to come up with some solutions, like the general solution to help those farmers actually losing their laborers and maximizing their productivity by using this technology. So again, this is a collaboration with the collaborative efforts between A&M University here in Corpus Christi with the Texas A&M Agri-Life Center in West Laco and Corpus Christi. The main goal for this project is on the autonomous harvesting for delicate crops like tomato, strawberries, and raspberry and stuff, and also the resource management is on one of the big goals because we try to improve the productivity of the farmers on the field, minimizing irrigation, nutrition, and disease control as well. The third goal is to increase the resilience to the impacts of the climate change and also the unexpected uncertain situations like COVID as well. So those are the main goals that we want to target on. And under these goals, we have some detailed objectives. So again, the first objective is to develop a remote sensing technology for crop monitoring in farm fields. This has been done for the last 5 years with the one of our teams in A&M Corpus Christi with the Agri-Life. I'm going to share some of the result from there. And also another objective is to develop AI-based crop identification technology. I'm going to discuss why this is important. And third one is crop yield prediction, because our farmers wants to see what would be expected yield at the end of season with the current growth situation. So we want to provide them some, the more tangible result at each time period during the whole season, so that they can manage their resource to maximize their productivity. And also the last objective is to develop a sustainable and autonomous harvesting platform. So we're going to share some of those recent research results. Well, the thing was when we started this whole project with the A&M Center, Agri-Life Center, well, we found that we are not using the same terminology. Basically, it took us for a while to understand each side, because of the fact that we are using different terminology, different approaches, and also the main research interest was a little different. So it is hard to shake hands at the first time, but we eventually had both sides, I think, was very patient to understand each other. We came up with some kind of agreement, okay, not to cross each other's line, but we try to understand each side's interest, and then the approaches. And also, it took us a little bit of time to understand terminology, but once we get to the point, we were able to accelerate and then put some momentum on this research. So I'm glad that we found a good team to work together. And we are still a good team also, I guess. So this picture basically demonstrate what is kind of a future goal that we want to achieve. And then in order to do so, one of our team in A&M University actually did a lot of work, and I'm going to share those work at the moment right here with the 3D what I call it surface 3D surface from motion. So UAV actually take all the pictures, and then as you can see, we can generate a 3D dimensional cloud point, and we are going to use this as a digital twin so that we can collect some samples and data for the AI training purpose, and also plus path planning purpose of the mobile platform. So I think this is a very important key success as a result of collaboration between engineering teams and then the agriculture teams. So let me close this from here. So again, with this type of data, we want to achieve that to minimize the research and maximize the productivity by identifying those areas that need more attention. And we try to deploy our robotic system to that location. And that not only just providing the irrigation, but also we can probably manage the nutrition supply and then disease control only for those areas that is needed. So that's basically our goal. Again, to maximize productivity, minimizing the resource. All right, so how do we do this? Currently what we do is to achieve those kind of a short-term objectives by, again, applying this remote sensing technology with the UAV to taking those photos and then putting this all together to create a 3D digital twin. And then we make a small mobile platform that is able to navigate on a very tough terrain like this. Because if you go out to the field, most of the farm field is very rough. So we have to make a platform that is able to adapt to those. So at the same time, we need to be able to control this robot with the GPS. We use DGPS RTK technology so that we can control the global location of this robot up to 10 centimeter. Because of the fact that the robot has to go through all this empty space in the farm field for monitoring and then plus harvesting activities. So that's what we're doing, focusing at the moment. So as you can see, Jack told me to turn off this noise, but I don't know how to do it. Anyway, so I just demonstrate the capability of adapting this robot on a very tough terrain like this. And then with this technology, we achieve also the navigating this robot with a very precision localization with the DGPS RTK technology on our campus, this demonstration here with our students. So we have both sides done from the perspective of our navigation and then terrain, basically navigation and then precision control as well. So those are the main achievement we made so far in last few years. In current stage, if I want to share the current stage, well, the global localization is pretty much done now with RTK-based DGP technology, achieving 10 centimeter accuracy. And then rough terrain navigation, we still need a little bit of improvement, but that the rocker bogie mechanism seems to be the best to navigate on the rough terrain. And ongoing research on this side is to add some multi-degree of freedom manipulator to basically to achieve a lot of different objectives like the control, disease control or harvesting or taking photos and stuff. And also we study machine learning approach to control this path planning of the multi-degree of robot, not only for the harvesting, but also for the health monitoring of the plants. So those are the current stage in terms of the navigation technology. And then this one here actually shows harvesting. For the harvesting technology, we want to add some, we are trying to add more on the 3D sensor-based crop identification and then temporal stage prediction. Those are, I'm going to share some of those results at the end. And also we are trying to basically develop some kind of machine learning based robot manipulator control. And then we use a reinforcement learning, and specifically we use DDPG, deep deterministic policy gradient technology, plus probability-based motion planning. So we try to merge this together to control the 6-degree of freedom robot in the farm field. Because if you think about it, it's very challenging, because this robot has to work very close to the farmers sometimes, and then farmers, because the robot may not be able to do everything, and people probably have to work together. We call this as a collaborative robotics system. So in order to do so, robot has to be able to avoid collision with the humans, because nobody wants to be touched by this robot. At the same time, robot has to be able to access to those fruits and then delicately harvest those. And how do we do this? So there are a lot of conflicting objectives, and we found that machine learning approach seems to be the best at the moment to achieve all these conflicting goals. So what we do right now is to test our robotic system in our lab to see if we can teach the robot easily, kind of intuitively with our gestures, instead of using teaching pendant to control all this position. We developed the 3D sensors and test on the robot, where we can actually just show the path that we want to go. And then robot basically will learn naturally with a human trajectory. And then we use this data to actually train our robots with this DDPG basics. So I'm sure some of you guys heard about this type of technology, reinforcement learning. Is just simply multiply connected network, which basically will simplify the what I call it, the prediction of the mathematical model, nonlinear model with this neural network system to predict, to generate the best action for a given state. So basically, for a given state, a robot will take a look, or this AI will take a look, what's the current action in the environment. And then by using this neural network, it's going to generate best action to take on this, on those situations by using this learning process. So we collect those data in the deploy memory, and then we use this data with the Bellman equation to train the robot. So again, this is a very simple schematics of the DDPG, and we try to somehow improve this technology to achieve conflicting goals. Because if you want to move to the right, and if you want to at the same time want to the left, how do we teach or train this robot to, and then how do we judge which one is best? So those are the kind of a question that we are trying to solve with our RL technology. And then as you can see, you can think of these mobile blocks as farmers walking around in the area, and then you can think of this blue as fruits the robot has to harvest. So again, there are conflicting goals here. You have to avoid those the farmers around. At the same time, you have to be able to approach those the fruits. So how do we do this? Well, we try to use multiple network, not only just one DDPG, but the multiple DDPG that is trained specifically for each task, and then we try to put together with a top layer. I'm going to show you the result. But again, that's where the challenge is on at the moment to achieve all this harvesting process. So I'm just trying to demonstrate harvesting is a very complicated task. And then the AI will actually also have difficulties to solve this problem as well. So currently we are trying to make some kind of a, what I call it, the digital twin lab in our school to teach this three-dimensional manipulators. Again, the blue one is the fruits, the red one is obstacle or the farmers around. So basically, this guy is struggling to learn how do I move my manipulators around to achieve the goal. So this is still ongoing research. And here's a kind of a quick architecture that I want to share, where current architecture we have right now. So here we have a path planning agent, which is trained to access those fruits once it is identified. And then the one on the right is the collision avoidance agent, which is trained to avoid any farmers next to in my workspace. And also, because those are conflicting goals, we try to put together all this into a hierarchical structure with the top layer here. And this top layer is going to take a look at the same environment where the state is defined, action is given from these two agents. And then this environment will generate next state, T+1, and then R. R is a reward system used for Venn equation to solve. So with this process, we actually generate different reward functions for each individual agent. So top layer, this guy is also an agent. And then the path planner, and just like the path planner, and then the collision avoidance agent. So each agent will receive a different reward to be trained. And then we try to use this kind of multiply, what I call it, deterministic reward functions to train this top layer. And what this top layer does is to observing this environment and take a look, okay, which agent I have to trigger, which one agent I have to trigger to achieve my goal. So that's what top layer is struggling for. And we are not, to be honest, successful yet, but we are pretty close, getting close. And maybe next time I can share some result out of this research as well. Okay, so eventually what we are thinking is, well, maybe the generalized AI, there are many different approaches, but generalized AI might be structured like this, like the brain. So for example, for human side, you can think of this top brain as your desire. And then at the bottom, right, in the second layer, you can think of this as, okay, I want to go home to sleep because it's too much of work today. And then second one, well, still I have to work because, you know, I have to write a proposal and I have to write a paper and stuff. And so I have to still work. So there are many different conflicting agents right here at the bottom. And then these has to come to the more detailed agent that allows you to write a good proposal or write a good paper. So these are the well-trained, for example, the agent in a very low level, detail level. But I think that this seems to be the kind of architecture that we have to pursue to make a generalized AI. So that's what we are studying at the moment with our graduate student in our school. Okay, so that was pretty much on the side of the harvesting with multi-degree of freedom manipulator. But what about the prediction, yield prediction? Basically, our farmers wants to develop technology to be able to predict what would be my yield for each individual areas in my farm. Because if you think about the huge multi-acre farms, all the situation is different. For example, the irrigation is different, nutrition may be different, and then the condition is different. So they want to see what is the best treatment to each side. So that we want to develop an AI that is able to navigate throughout the whole field, capturing all these images and data, and generating a kind of a map of proposing, okay, you have to go here, this side to control disease. You have to irrigate more on this side. So that's what we are trying to do at the moment. And we try to achieve this with the two AI agent. One is AI breed classification, and the other one is AI-based temporal stage prediction. Well, the reason why is because each breed, for example, that we are testing with tomato at the moment, but there are so many tomato breed. Each breed has different yield with different weight, and everything is different. So first of all, the robot has to understand what kind of breed I'm dealing with. And the second of all, we want to know what is the growth stage of each plant by looking at based on sensor data, for such as camera image or 3D cloud points. So we are trying. We are at the moment. We have been successfully created AI for breed classification, and also AI temporal stage prediction as well. So I'm gonna share some of those findings that in this course of study. So CNN-based image classification was the main target for this breed classification, and if you think about the tomatoes, they are different breed, but it's very hard to tell which one is which by our bare eyes. So how do we do this with the AI? Well, that's a very big challenge. But we put together a TensorFlow and Keras technology to create a CNN. And this AI has 2 CNN and 2 fully connected layers. And we achieved about 82% of classification accuracy. We tested 5 different breeds of tomato, and this AI can tell, okay, this image belongs to what breed, up to 82% accuracy. Second one is, again, temporal stage prediction, meaning that, okay, what would be the stage of growth? Is this a 2nd week or 3rd week or 5th week? Because we want to see how much of the research has been applied, so supplied on this plant. So same approach is taken, CNN-based growth stage prediction technology. The challenge was for this particular task, we have a very extremely small data size. Our graduate school student went to the field, the house, the indoor house, every week, but only capturing like 20 to 30 images every week. So it's going to be really extremely small data size. And we tested transfer learning to see if we can somehow train these well-developed ResNet or VGG, but it was not satisfactory. So we come up with our tailored design with the 3 CNN 2 fully connected. And then we achieved up to 85% accuracy on this as well. So this is a picture of showing some of the photos we collected. This is, again, West Laco greenhouse with a cable robot right there. So this cable robot is installed and at each row right here, and then it basically have a camera at the bottom and moving and collecting all these data points. So these are data we collected, and then the label is also provided at the end to train these images. So again, this is kind of quick pictures of the 2 convolution layer and then 2 fully connected layer made in Google Colab and TensorFlow. So this allows us to achieve, this shows us how do we actually increase the number of image by using data augmentation. As you can see, here's the plant right here. Each plant is right next to each other. So we have to divide these into pieces. And then each plant has a label right there. These are human. We just wrote down and then put them on the floor so that the robot can take all these pictures together with this label. And then we did several level of data augmentation and had about 2,400 files. And we trained them with 75% of this and then used 25% for validation. Here's the result for the breed classification result, going up to 82% here for the train accuracy. And then validation accuracy, it was pretty close to 100%, about 90%. And then it shows also the train loss and the validation loss going down, so that it basically shows that this CNN converges with some meaningful accuracy right here. And this is the chart that shows us, okay, well, I've seen that a lot of domain experts that was wondering, okay, how many data do I have to collect to train my AI? Well, we did some basic study on this, and then it shows that, okay, there are some threshold that you have to go for example, in this particular task, it shows that the 300 number of images seems to be enough to get to the point of 90% accuracy, and it doesn't grow too much. The accuracy do not improve too much after this 300 number of images. But you have to make sure that you basically come to this point. But if you set your goal, for example, okay, well, 80% is okay for me, then maybe 200 image data number of images should be fine. But again, this is one of the specific tasks that we did, breed classification. So depending on the application, this graph may change, but what I want to show you is that there is kind of a maximum number or a minimum number that you have to collect to train your AI. And then at some point, you do not have to collect more to improve accuracy. So that was the result of the breed classification. But this one here is for temporal stage classification. This is the greenhouse in the Corpus Christi, and we did the same thing over here. But this time we actually take photos manually instead of using a cable bot, because of the we try to focus more on the very delicate growth stage of each plant. And here we actually measure the weight and then temperature and image. Again, all these are installed by the AgriLife Center, A&M AgriLife Center. And then we also controlled irrigation. One side is 100%, the other side is 50% irrigation. And we try to use those as a validation data after we train this AI. And also we collected temperature. So with this, as you can see, this is the, I think, 4th week, number 9, 5th week, number 3. So these are the examples of the tomato. And then we had about, I think, 5 different tomato breed as well in this case. But the challenge is that the very small, extremely small data size. As I said, we just collected for the first 7 weeks because the plants are pretty much grown now after 7 weeks. And then there are only 27 data sets in each week. So we have only like what, 150 roughly data sets, which is not even enough for us to train a CNN. So we tried to train ResNet and the BGG, but it wasn't satisfactory because of the small data size. So what we did was, again, we applied data augmentation again, and we found that the random crop and the random translation in June, these seems to be working the best out of those all the trial. And then we come up with this particular the structure of the CNN, 3 convolution layer and then 2 fully layers. And then we used some dropout to randomize because of the small data size, and we achieved about 80%, close to 80%. So what this means is that if you show an image of tomatoes to this AI, it will be able to tell what is the growth period. This is a 2nd week image. This is 3rd week image, and this is a 4th week image. So with that image, capability, now we can put together a kind of yield prediction system. So as you can see, we provide an image like this, and then we also provide growth period, so which is the ground truth of that growth period. And by using these 2 data, the AI will actually try to figure out the temporal stage of that particular image. For example, in this case, this guy says, okay, even though the image is taken at the 4th week, it seems like it is taken from 3rd week. That's basically what it says. There could be a lot of reasons. The one reason would be maybe the irrigation was not supplied 100%, or there are some plants suffering because of the disease, or because of temperature, all that stuff, a lot of different stress. So that's basically what AI can tell now. And then also this guy can tell us what kind of breed you are looking at. So the reason why we need the breed data is because we know that the maximum possible yield of each breed in our database so that by looking at this database, we can say, okay, with this 80% of irrigation, you can achieve up to a certain amount of the yield at the end of this season. So that's what this guy can do. And at the same time, because it could tell, okay, well, you have only 80% irrigation, so you have to apply more to maximize. From now on, you have to apply more irrigation at that point for this particular plant to achieve the best amount of the yield. Okay, so that's basically what this whole thing is designed for. So we use some AI technology, and then we use some interpolation technology to predict yield, and then to recommend extra irrigation. So here's some testing experiment. This is again the plant with only 50% irrigation, and the AI. After we train this AI, again, if you see all the ground truths in the second row right here, this is a randomized testing. So this says ground truth is a 3, 30 week, first week, 5th week, 5th week, 4th week, all the way going along like that. And then the AI told us, okay, well, this image seems to be the first week, 3rd week, 2nd week, 1st week. So again, it's not correct, precise, not really precise, but it is able to tell how much of what seems to be the growth stage for the given 50% irrigation data. So for example, this guy 3 or 2, this really seems like very close to each other because everything is integerized. And the average accuracy was about 54%, meaning that, okay, well, for this particular image, looks like you only apply the 54% of the irrigation. You need apply more if you want to achieve more yield. By using this interpolation equation, you can actually predict the I, which is the predicted irrigation. I'm sorry, not the yield, but the predicted irrigation. So with this number of irrigation, we can recommend to the farmers saying, okay, you need 20% more, 30% more of irrigation on this particular plant. So almost done now. So closing up, what we want to do, what we have been achieving so far is that we are focusing on the harvesting technology with the robotic system, applying the RL, reinforcement learning, machine learning technology on that. And also we apply the CNN technology for monitoring the plant growth stage plus the breed classification. So, well, still we have difficulties on collecting those data by considering those huge farm field areas and then the acreage. So what we want to do is to develop a very precise digital twin, not only the 3-dimensional, the digital physical twin, but also the gross model that basically can grow digitally, those plants grow digitally with the given supply of nutrients, waters, and disease states and all that stuff. So if you apply all this data, we want to be able to create a digital model like this. And then we can create, with this model, we can create a synthetic farm field where you can actually put these model in each row with different growth conditions. And then with this, what we can do is to collect the data, training data, very easily. Instead of going out to the field and taking those actual images, we can actually use this synthetic farm field to collect the training data, because collecting data is always a big challenge in AI training. So with this, again, we try to accumulate 4D vector space. 4D means, okay, 3D plus time, so that we can accumulate all this data and then train this into the AI, so that AI can grow this digital twin model digitally in the computer system, so that putting together all this, we create this static farm field. Okay, now then with this static farm field, it will facilitate our training data collection, and also at the same time training AI to bring it up to the next level with more accurate capabilities of predicting the yield and stuff. So again, the future study with this achievement so far, we are going to work on disease and then disease classification and nutrition supply, not only the irrigation, but we are interested in how do we predict the level of nutrition supplied on this plant so that we can create very accurate and precision yield prediction model to help our farmers to maximize their productivity. Well, let me wrap it up like this. And I'm sorry that I have some other meeting, but I can take some questions if anyone has for about 5 minutes.
Okay, thank you very much, Dugan, for a very interesting presentation and very powerful how AI and robotics can help. Now, my understanding is that most of your focus is on applying this at the commercial level. Do you also see applications for research? And one thing that a researcher would want to do, for example, at the time of harvest, is take all kinds of measurements on the individual tomatoes. Are you thinking of those the possibilities?
Well, I would call myself as a researcher, not a commercialization agent expert. So okay. We're trying to help farmers. I mean, to see if they understand the whole process and they try to understand how do I maximize their productivity. But what we are doing here is, again, just doing a lot of studying research. And possibly, yes, I mean, if there is a chance, we can commercialize this technology, but we understand the basic level of physics behind the biology. And then we try to put together those into a digital twin system to make a kind of a growth model, precision growth model. So, well, we are doing still on research stage, but well, if anybody is interested in commercialization, yes, why not? We can talk.
Yeah, I actually, and I'm sorry, I wasn't very clear. I wasn't talking so much about commercialization of the technology, but your target is the commercial farmer, developing methodology. And
for researcher, yeah.
That the commercial farmer can use. But how applicable is this to a researcher, a plant researcher?
Research society, yes. I'm sorry, but again, for research society, we can. Our goal is to, to put together information that we collected and then research result and publish those. Again, those publications will help them out, understand what we have done. But anybody interested, we can share the AI model that we developed so far, and then I mean, these are not proprietary or any, there's no, the IP test on it. So I'm okay to work with all the researchers for the collaboration, and also these tools that we developed eventually will be probably online, so they can actually download and then they can test to see if they can apply this on their own research areas.
Yeah. So are you working on ways to, as the robot harvests fruit, to take specific measurements on the fruit?
Specific measurement?
Yeah, like the weight of the fruit, and then tie it to the plant, or the color, or any defects.
Well, at the moment, as I see this picture again, as you can see, these plants are hung over on the ceilings with the weight, the gauge on top, so that we actually measure the entire plant. And then eventually, as you can see the fruits right here, we should be able to measure the fruits as they grow. I mean, the robot has to be able to see those fruits, and they should be able to identify the colors to approach, to judge what is the best time to harvest as well. So measuring and monitoring is one of the big issues for harvesting. And then at the moment, we use this data in the indoor warehouse to monitor the growth stage of each plant. Not only just the plant, but also we actually use we actually measure the irrigation amount as well.
Yeah. Okay, one last question from the chat, because I know you have to leave. Is everything you have worked on only RGB, and how would you collect or sense data to help you train a model to identify something like nitrogen deficiency?
RGB, yes, we use RGB, but we use two different cameras. One is RGB, the other one is infrared. Infrared gives us more information when basically the light is not really friendly. So we try to use both. It seems like RGB seems to be helpful at the moment because of the extra information added on to classify the data. But as I said, we try to apply augmentation to more generalize this AI to deal with daylight outside. So that's a good question, and we are still working on it. But all the work we have done so far is from the image in greenhouse. It's a well-controlled environment. Well, I mean, the daylight is changing, so that's why we actually applied some augmentation, light augmentation.
Yeah. Okay. Thank you very much, Dugan, and I know you have to leave.
Thanks so much.
So thanks for your presentation, and I'm sure people will contact you if they have additional questions.
My pleasure. Okay. Yeah, if you want to talk to me, just here's the email. So let me know. Anybody?
Okay.
Interested.
Thank you.
Thanks so much. Okay. Bye.
Yeah. Thanks. So Edwin and Anthony are still here, and so there was another question in the chat. Does your method or software work for a large cornfield where there are no aisles between the plots or grids? So, for example, the plots are 30 by 30 meters. Would the software work for that kind of a situation?
Thank you, Jack. It's a great question. Just checking, you can hear me, right?
Yes.
Okay, great. Quick question. Would it be okay if I share my screen just once more so people know how to contact us later if I can't answer all these?
Yes, that's fine. Yeah.
Okay. Thank you. Thank you. Just a moment. Okay.
Yeah, and I see that
all right, so
Edwin has also put in the chat a link for contact.
Yeah.
Go ahead.
Thank you. Thank you. So yes, that's a great question. So for like a large or continuous cornfield rather than something that's planted in straight rows or plots. So Phenix has, at least our software, it's got limitations. The stitching algorithm is really designed to stitch imagery of small plot trials. So it's really optimized for that specific application. If you've got a field that's truly just continuous corn where the seeds are just randomly scattered and there's no like visible row lines or anything, I don't think our software would work very well for stitching that kind of terrain. And to be honest, most software that I have heard of will struggle with that type of terrain. But let's just pretend hypothetically we could stitch something like that. Then yes, there's no reason why you can't set up like a 30 meter by 30 meter grid over the field and think of each, grid unit as like, rather than a plot, just think of it like a measurement zone. And you can, extract those measurement zones from your original photos and generate metrics for them, just like you could for a small plot field trial. The catch there is, again, our focus is on small plot field trials. So our workflow is assuming that your plot size is small enough to fit inside of your original photo size. So the bigger you make those plots, the higher you have to fly to make sure your image footprint actually captures whole plots, because that's where we pull the data from. So I hope that answered the question.
Okay, thank you very much. Yeah.
I'll just say one last thing, please. If you would like to talk to us more, if we can't answer your questions, visit our website at plotphenix.com. It's really easy to book a meeting with us at your convenience.
Okay, thank you.
Thank you.
So I think we have to wrap it up here, almost at the top of the hour. So thanks, Edwin and Anthony, for your contributions to this field day, and it's been very interesting to hear both of these examples. Just a couple of things. So Nicole has put in the chat a link to a survey, an exit survey. So please, as you leave, fill in that survey, complete that survey. And there's also a Slack channel that we can use to continue the conversation if there's additional questions. But yeah, feel free to contact either of the two groups directly also. And a reminder of field days. The next one will be December 15th, the third Wednesday of every month. So look for that. There on December 2nd, there's a training workshop on structural variation from 1 to 3. So look for that. And then there will be another community survey for us to get more feedback from you on what the needs are, what the challenges are, so we can inform NIFA. So check your inbox, or if you don't see it, then just email Nicole directly to receive a link. So hopefully you'll take the time to do that, to provide that valuable information. Always go to AG2PI if you need any information. Everything should be there, including the recordings. And with that, I just want to thank also Nicole Scott for all of her help in making this possible and making all of this work. And thank you for attending, and we'll see you hopefully next month. Thank you.